The Long Island Horticultural Research and Extension Center is a campus of Cornell University. We've been here in Riverhead, New York for 98 years, and our purpose here is to help the agricultural industry succeed. There is a lot of need for up-to-date research and assistance with growing plants. The impatiens is primarily of interest to the greenhouse growers on Long Island, as well as some of the nurseries, the commercial, the retail establishments that sell plants. Right here we've been breeding impatiens plants, trying to develop resistance to the disease called Impatiens Downy Mildew. And there's always problems when you do breeding, and one of the issues that we found out with this plant is they have what we call explosive seed pods. And it means when the seed pods are ripe, they explode. And of course, if you're trying to harvest seed, that's a problem because then they're all over the greenhouse floor. So we use a technique called plant tissue culture to assist our breeding program. And essentially, plant tissue culture is a way to grow plants in a sterile environment on a nutrient medium under a controlled environment. So we will remove the pods that we've pollinated on these plants about 14 to 16 days after we've pollinated them. And we take those pods to our laboratory. They're sterilized on the outside, and then they're opened aseptically to expose the seeds. And those seeds then are placed in tissue culture to germinate. The nice thing about that process is that once they germinate in tissue culture, they're always going to be there as a stock plant for us. So they're always going to be available. We can take cuttings of those plants, bring them out here in the greenhouse. If we find out that we have a successful plant, a successful hybrid, we have the mother plant in tissue culture, free of diseases, free of insects, and we can then rapidly propagate that plant to increase the numbers quickly. And these are the results of some of that breeding research. So all these plants that you see here are resistant to the disease. We've done field trials for them for the last 2 years. But our next step then is to produce a plant that has a nice looking flower and a nice symmetrical growth habit. So we are continuing to hybridize these plants, and we'll continue to develop until we get a nice seed propagated line of plants.